following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
And what I want to do next is I want to look at Penet grids, because that's a very useful genetic tool. I just introduced it in the monohybrid cross we just did. I wanted to do another example so you can have or develop a much better understanding of how to use this very useful genetic tool. So manipulating a Penet grid is a very basic skill, and it's one that you should definitely look to master. Now, the example on the screen states that tallness is dominant over shortness in pea plants. A homozygous tall plant is crossed with a short plant, and we're asked to do two things. So using a Penet grid, determine the genotypic and phenotypic makeups of the F1 offspring, and the second part of the question is perform a cross of the heterozygous F1 offspring and show the resulting phenotypes and genotypes of the F2 offspring, the second filial generation. Okay, so the first step So in the first step, what we are going to do is we are going to determine the alleles that are in the gametes of each parent. So we've been given the parental genotypes. So we've been told one of the plants is homozygous dominant for tallness. So we're going to use the letter T, or the capital T, to delineate the allele, the dominant allele for tallness. And because it's homozygous dominant, it's going to have two of those. And we've been told the pure breeding dwarf plant, which it's crossed with, is homozygous recessive. So it's got two of the recessive alleles denoted by a small letter T for dwarfism. So this is our cross right here. We're crossing a homozygous dominant variety with a homozygous recessive variety. So according to Mendel's law of segregation, each of these alleles will segregate into gametes separately from each other. So that will segregate into one gamete, that will segregate into another gamete, that will segregate into another gamete, and that one will obviously also segregate in a similar fashion. So what we have right here, these are all the alleles or the gametes we'd obtain after that segregation has occurred. So now that you know all the possible alleles that you can get from each of the parents involved in the cross, in the second step, we're just going to take those and we're going to place them on either side of the Penet grid. So for instance, if the tall plant was the male, putting those two alleles at the top right there would look like that. And if the other plant, the recessive dwarf plant, was a female, would put those two alleles on the side right there. And all we are going to do in step 3 is we're simply going to cross or combine these alleles to obtain all the possible allele combinations of the F1 generation. So that cross right there would give us heterozygous genotype. That will give us another heterozygous genotype, crossing that and that, yet another. And finally, at the bottom right there, another heterozygous genotype. So as you can see, all the offspring in the F1 generation, the first filial generation, all have a heterozygous genotype, and they will all obviously be tall because of the simple fact that the allele for tallness, the capital T, is dominant over the recessive allele for dwarfism. So all the F1 offspring will be tall. This is just like we saw in Mendel's experiment where all the F1 offspring were purple colored. Now, in step 4, to determine the genotypic and the phenotypic ratios in the second filial generation, what we are going to do is we're going to repeat the exact same process. So we're self-pollinating, we're cross-pollinating F1 hybrids. Now remember, all of the F1 hybrids have this genotype. They have the heterozygous genotype. They're all capital T, small t. So if we crossed a male F1 hybrid with a female F1 hybrid, which obviously has the same genotype, the gametes that we get from that cross would be as follows. So these are all the possible gamete combinations that we could get from that cross because they're all heterozygous. So we're going to take those possible gametes and we're going to put, for instance, if we had the male at the top right here, we're going to put the capital T right there and the small t for the recessive allele right there. Similarly, for the female, on the left side of the grid, we're going to put the capital T right there and the small t. So what we're going to do next is simply cross those alleles to get all the different allelic combinations that are possible in the F2 offspring. So that would give us capital T for that one, and that would be a homozygous dominant genotype, and then we'd have a heterozygous genotype right there. And the third cross at the bottom left-hand corner would give us another heterozygous genotype, and the last cross would give us a homozygous recessive genotype. So as you can see, things have changed somewhat in the second filial generation. So if we're talking about the genotype, we have three genotypes that we are seeing in the second filial generation. So we have the homozygous dominant genotype right here. And so we have one of those. And we have the heterozygous genotype right here, 2 out of the 4. So 2 of those. And we also have the homozygous recessive genotype right there, 1 out of the 4. So that's 1. So the genotypic ratio in the F2 generation, the second filial generation, would be 1 to 2 to 1. So 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So how would these genotypes look like at the phenotypic level? Very simple. Like we said, the allele for tallness is dominant over the allele for dwarfism. So this this offspring will be tall, this one will be tall, this one will be tall, and the only one that will be short will be this one right here. So this is exactly what we witnessed in Mendel's cross, where we had the white color disappear in the first filial generation and then reappear in the second filial generation. Similarly, in this case, the short or the dwarf plants disappeared in the F1 generation, and they are reappearing in the second filial generation. So our phenotypic ratio in this case would be 3:1. 3 of them would be tall, 1 of them would be a dwarf. So using a Panett grid, we have very easily described or shown what is a monohybrid cross, which basically explains Mendel's law of segregation. So these alleles are all in gametes that's segregated independently, that's segregated from each other and found themselves in individual gametes, either in an egg or a sperm, depending on whether you're talking about a male or a female. Now,